In this screencast, we're going to discuss the cell membrane and basically its structure and overall function and the role that it plays in keeping a cell alive. Just so that you know, like always, make sure that you complete the assigned readings and also, like always, seek out good, accurate resources that are available to you. So first we'll talk about probably the main, or at least most numerous component of the cell membrane, being the phospholipids. And remember that phospholipids, we talked about these with the other macromolecules earlier, that phospholipids have a kind of a two-part to their molecule here. The phosphate head of it would be hydrophilic. Remember that it would be attracted to water because of its polarity, because of its slight charge. The fatty acid tails, those would be hydrophobic, so in other words, repelled by water. So it makes up a pretty good cell membrane when it's arranged in a bilayer, two-layered here, and simply by the orientation of these molecules to one another, the individual phospholipid molecules, it's going to set up a barrier. The phosphate head is going to be attracted to water or to an aqueous solution, and the fatty acid tails are going to be repelled by water. And simply due to that nature, it's going to set itself up as a bilayer. So again, this is really simply due to the characteristics of the phospholipid macromolecule, the charges involved and the water characteristics that it holds. So again, the hydrophilic heads would be polar and the non-polar hydrophilic tails. And so they're going to align themselves in this way. For those reasons, the cell membrane, the bilayer, is going to be impermeable to polar molecules. The non-polar tails are going to be impermeable to the polar molecules. Again, so the orientation of this phospholipid bilayer, this lipid bilayer of a cell membrane, is going to become a non-rigid, so it's going to be flexible and really non-permanent barrier. In other words, it's not like a brick wall that is static and put together with mortar. It's a fluid membrane. More on that a little later. So the number one job of the cell membrane is this traffic control. It's the transport. And we'll talk about this traffic control a little later, but it's going to control what goes into and out of the cell. In some pretty complex methods and manners, and in others some pretty simple ways. But the cell membrane is going to separate, it's going to compartmentalize a cell, which again, remember, that allows chemical reactions to take place in a tight location, makes them more likely. But it controls the traffic in and out of the cell, transport across the cell membrane. Remember, it's not simply structure. That's the job of the cytoskeleton. If we're talking about transport across the cell membrane, then we're talking about a characteristic called permeability. A cell membrane is going to be semi-permeable, or more accurately, it's going to be selectively permeable, kind of like a screen on a window or a coffee filter or any filter for that matter, or a strainer or a colander. Some things are able to cross or get through and some things are not. Much like the key and lock or the induced fit model that we talked about with the enzyme substrate situations, specificity is an important characteristic of some of these molecules that are embedded in the cell membrane. So there's more involved than just the phospholipid molecules. Another important compound or another important molecule involved in plasma membrane would be cholesterols, and the actual fat composition is going to affect how flexible or how fluid it is, being the plasma membrane. And of course, just mentioning here, you want to go to your sources and look at how the cholesterols and the types of fat composition of the membrane are going to affect its fluidity and flexibility. Also, arguably more important that is a part of the cell membrane are the proteins that are embedded. A transmembrane protein. transmembrane. That means it's going to go all the way through the cell membrane itself. This helps create the semi-permeable channels. The reason is that these things, the transmembrane proteins, often will form a channel that will allow some substances through from one side to the other. Although it varies on what specific type of cell you're talking about there's about one protein for every about 25 lipid molecules. So they're pretty numerous in most cells. So what's the importance? Basically, what's the importance of these proteins and the role that they play as being really members of a cell membrane? Something to keep in mind is remember that protein molecules are built from amino acids that are bonded together to make a polypeptide chain, or to make a long chain, otherwise known as a protein. Of these amino acids, some amino acids contain non-polar R groups, and some amino acids contain polar R groups. Well, non-polar would coincide with hydrophobic tendencies, and the polar molecules would coincide with hydrophilic tendencies. So what role do the proteins play in membrane transport? Again, proteins are built from amino acids, and if you take a look at these particular non-polar amino acids, they would be non-polar and hydrophobic, so they would not align with water. In these examples, you've got polar amino acids. In other words, they're going to have that slight charge to them, and that would mean that they would be hydrophilic. Well, what role would that play in what type of molecules may cross the cell membrane? It's the arrangement of these amino acids in a membrane protein that determines whether the membrane protein will insert into the nonpolar lipid bilayer and how it will be positioned. So within the membrane, you've got nonpolar amino acids that would be hydrophobic. This would anchor the protein into the membrane. On the outer surfaces of the membrane, in the fluid, the polar amino acids would be hydrophilic. So they would tend to extend into the extracellular fluid and into the cytosol, or into the cytoplasm. So they would extend into the environments that are outside of the actual cell membrane environment. So just a couple examples here. You've got aquaporins that are water channels that allow water molecules to move freely across a cell membrane. You've got proton pumps that, again, are set up due to certain characteristics of these amino acids that make up the proteins. You're allowed to pump ions or protons, specifically across membranes. That creates a concentration gradient. More on that a little later, but it can store potential energy for later use, kind of like a battery is able to store energy. But it gives us an idea of how molecules that are in a cell membrane, they help transport molecules or substances across that same cell membrane. So there's a lot of specific functions that we're going to address later. Many functions of the cell membranes. They can be transporters. They can deal with enzyme activity. Of course, you look at a substrate enzyme complex here with a specific product to help chemical reactions take place. They can be receptors. Again, like the specificity we talked about before, where a key and a lock have to match up. Identity markers, much along the same thing. This is how they recognize how they may recognize, for instance, an antigen, a specific protein in the environment. They can help cells adhere to one another to make up tissues in an organism. They can also attach to the inner workings, the inner structure of the cell, in the cytoplasm, or called the cytoskeleton, that is going to function in maintaining a specific shape. There's also signaling that we'll get to. Signaling, transmitting a signal from the outside of the cell to the cell nucleus, which is, for example, maybe a particular hormone is going to trigger the cell to produce a particular protein so that it can be released. All sorts of different functions of the cell membrane. So again, the proteins are really important. They're going to determine the specific functions of that cell membrane. The cell membrane and the organelle membranes each have a unique collection of those proteins, unique to whatever type of cell it might be. We've got both peripheral proteins and integral proteins. The transmembrane proteins that we talked about before are going to extend all the way from the internal environment to the external environment. They typically do a great job of helping with transport, specifically through a channel. Peripheral proteins are typically going to be some sort of identity marker, almost kind of like a bouncer at a door that's going to recognize specific markers and then proceed to decide how the cell membrane is going to respond to those specific markers. As we started to mention before, this cell membrane is much more than just the lipids, and we talk about its structure being a fluid mosaic. Again, that specific fluid mosaic model takes this entire structure, dealing with the hydrophilic region of a protein and the hydrophobic region of the protein, that are going to match up with the phospholipid bilayers because of their properties that we already talked about, but it's far from just a rigid, unchanging structure. It's got fluidity. These molecules can actually almost float around and change their structure. This lipid is not locked in place. This protein is not locked in place. So its overall structure is very fluid. The extracellular fluid, extracellular, outside of the cell, versus the cytoplasm are going to be two different environments, and that's going to set up some what are called concentration gradients of substances that we'll get to a little bit more later. The carbohydrates that we talk about are also a part of the cell membrane structure, and these carbohydrates are going to be attached to either a protein or the lipid part of them. So those carbohydrates, glyco referring to the carbohydrate, you can have glycoproteins or glycolipids depending on what they are actually attached to. And these are always, typically involved in signaling. So these things are going to recognize certain substances that are very specific, helps the cell distinguish one cell from another. In the immune system, those specific cells are going to recognize foreign cells and identify them as harmful or not. And for instance, the antigens of red blood cells, the groups A, B, and O, they differ in their external carbohydrates that are on those red blood cells. Therefore, the body cells itself are able to recognize whether a blood type is the proper one or whether it's the wrong one. And if the antigen that we're talking about is the wrong one, the body will reject those foreign cells. So again, you want to understand the main function of the cell membrane. It's really to control transport. But the structure and the macromolecules involved and understanding of the fluid mosaic model can be pretty complex. So you want to, again, spend time with your readings and spend time with any extra resources that are provided or available to you.